I have questions. I've given them to Dr. Rust. I don't know if she's posted them on Blackboard, but hopefully she'll do it today or tomorrow.
Yeah, I don't know if that's gonna happen.
Really?
Yeah, usually takes about.
I was looking for him.
Okay, well, I gave them to her, and I talked to her. I'll see her today or tomorrow, and I'll remind her to do it. And now, don't freak out. I've done some lengthy stuff for you to really, if you get it, it's best, I don't know, it's best to do these in study groups so that, you can begin to discuss the material. But if you go through the study guide, I guarantee you, and if you've got this information down, I guarantee you should do well on my questions.
Okay.
Huh? You don't trust me?
Well,
I've been doing this for no. I've been doing this for many years, so I'm serious about that. No. We're different. No. Our approaches are different. But no, I've been teaching I teach this course. I've been teaching this course now, I don't know how many years. The second time. But maybe probably last 4 or 5 years. And I do M semesters, and they usually do pretty well on this exam.
I hope so.
Try to help you out. But anyway.
Yes, thank you.
Do the study guide, and we'll talk about. I'm not going to post the answers because I want you to do it. We'll talk about the answers when we come for the review session, because I've had review sessions and people come and they just sit there and look. And so I post a question so when you come with that study guide, you hopefully have done it, and then we can go over the answers. Yes?
We have one more class period after Thanksgiving.
The Wednesday after Thanksgiving is our last period. It's the last class. And then you have a Thursday review session for renals, and that's it. So after Thanksgiving, you got one class and a review session, and then a review session on Thursday. Friday is a study day. Okay? So I want you to, before you start eating the turkey, start trying to go over these study questions, and I'll remind Dr. Rust.
Are those the same questions that are just in the syllabus right now?
No.
So should we do the new ones, not these?
Which questions in the syllabus? The
The objectives?
Yeah.
So what does she mean study guide?
The objectives, the way the objectives should work.
So we should forget about the objective and focus on what she's going to give us?
Before you come to the class. Okay? Yeah, so you should have an idea of what we will be discussing. All right? So you've done the objectives?
No.
Oh. The objectives tell my books. I need to talk to you. You gonna be around?
No.
Okay. Okay.
Question?
Okay. So,
She has a question.
yes.
So your questions are sent back? The questions you providing are sent back?
I'm providing, in the study guide, I'm providing questions, straight questions. Now, don't expect me to take the same questions and put them on the exam. No. But the material, you should be familiar with it, and I can, if I adjust it a little bit, you should be able to pick it up readily. But there are a number of, points that I put. I think there are about 40-something questions that I put on the study guide.
Seriously?
All right. So, they're covering everything, repro and all of endo. Okay. So, male reproduction. So we talked about how we get the male sexual organs, internal, external. So let's just see, where do we want to start? Let's start with the bladder, which has nothing to do with the reproductive system. The only thing, this break woke some people up. The only thing start here with the bladder. And we know that that is what's holding the urine. And we know that the urine passes out through the urethra. Okay. But in the male also, you can see that you've got some interaction with the reproductive system and the bladder. So let's say here the testes are down here in the scrotum. And we said that within the testes, you get the sperm development. That's about 64 days. From the testes, we pass to the epididymides, which are these two little purple structures. That is where the sperm cells acquire their motility, and they stay there for about 12 days. So 64 plus 12, and the epididymides, and I'll show you how to spell that in a minute. Okay, now from there, they go up along the vas deferens, and that's where they're stored. They're stored along the vas deferens until either they're ejaculated or they degenerate. Now, at the time of ejaculation, then you can see that you're passing here. The bladder, this is not a really good diagram, but you know that the sphincter to the bladder must close. And as that sphincter closes, as the sperm are passing through, you don't want them to go into the bladder. You don't want them to be retrogradely projected to the bladder. You want them coming down where the seminal vesicles will add their fluids. And you want them coming down through where the prostates will add its fluids, the bulbourethral gland, and out through the urethra. So what we're saying, everybody knows that the sperm travels out the same path, exits the body through the same path as the urine. So this, keep talking about the bladder closing. You call that retrograde ejaculation if the bladder does not close. So if it does not close, instead of the sperm being ejected here, it goes into the bladder. So is that person fertile?
No.
Can they make that person fertile?
Yes.
Yes, definitely. What they would do in the andrology lab is to ask the person to go and urinate immediately after supposed ejaculation or after climax. And that take the urine and wash the sperm cells out of that urine, because you know urine is going to be a very hostile environment. And you can begin to wash them and deal with them in terms of buffering them and culturing them, and then later they can be used for artificial insemination. Now, the reason that I bring that up is because people that have prostate surgery, sometimes they can develop this retrograde ejaculation with prostate surgery. Now notice too, the prostate gland is only one of them, and notice how it's surrounding the urethra. And so benign prostate hypertrophy can reduce the urine flow, and can you begin to understand why if this enlarges? Yeah. So there will be just a slow drizzle of urine because basically you're blocking the urethra, right?
So when you cast somebody, you just like opening that lumen, or just like the prostate swelling, so it's good.
Yes. Okay. Now here we're looking at a cross section of the penis. And you can see a number of tissues here, the corpora cavernosa and the corpus spongiosa. You can see where the urethra is and the blood vessels and all of that kind of good stuff. So, a couple questions for you. What makes up the erectile tissue of the penis?
Cavernosa and spongiosum.
Okay. Cryptocorpidism is when what happens?
The testes doesn't go down.
The testes do not descend properly, one or both, okay? Because it could be one or two. The accessory sex glands, what are they?
Oh, Lord.
Prostate gland, bulbar gland.
Okay, so there are three: the seminal vesicle, the prostate, and the global urethra.
What's different?
The prostate gland.
Prostate gland and
Seminal vesicle.
It doesn't come into the urethra. This is not a good diagram. It's not a good diagram. So when vasectomies are performed, where are you snipping?
The seminal, the seminal, the ejaculate.
Right in there, both sides. The vas deferens. So if a person has a vasectomy, are they still producing sperm cells?
Yes.
Yes. But the problem is the ejaculation of the sperm cells, because they're going to be a buildup down here. And sometimes you can see what they call varicoceles, where there's a buildup of sperm cells and there can be like an immune reaction and the cells begin to agglutinate and they just clump it in there. And so sometimes that develops with vasectomy. And if a man has a vasectomy, yeah, they can reverse it. They call it vasovasectomy, but generally, from what I've seen, those sperm cells are, when they're ejaculated, they reverse that vasectomy. When they ejaculated those sperm, they agglutinate. They're just in one big clump. And generally what you have to do is to do some work in terms of buffering them and probably artificially inseminating a female. And that might be very expensive because it may not be just artificial insemination. You may have to inseminate the egg, the actual egg. Yes.
So, like, during an erection, is it hard to urinate because of the contraction of the muscles, or is it because the sphincter of the bladder is closed?
Bladders should be closed. They should be closed. Okay, so now we're looking at another view here. And of course, we saw the testes and then the epididymis, as I was telling you. This is where the sperm cells acquire motility, and they stay there for about 12 days. And from there, we're going here to the vas deferens, making this loop, and this is where the sperm cells are stored until they're ejaculated, disintegrate, or some of them just drizzle out in the urine from time to time if they're not ejaculated. Because remember, I said 300 million a day. Okay, so now we're turning this loop